All right, so now we are going to take a look at some of these virulence factors. So remember that virulence factors are something that's produced by individual pathogens. They determine the extent and the severity of the disease. And remember, we spoke about this when we spoke about molecular Koch's postulates. How if we took an organism and we can identify its genes of virulence or its virulence genes, those are the ones that are expressing the virulence. That then what we could do is we could knock out those genes or cover them up in some way molecularly, and then we can give that organism to a host, and then they don't have that disease. They don't have, or maybe the severity or the extent of the disease. And then if we then took organisms from that host again, and then we turned on the genes again, and then put them back into a host or another host, then they would again show pathogenicity. So these are the genes that are encoded and that we can identify. Now, when virulence factor genes are then inactivated, then we don't have that virulence anymore. And so we know that those are the virulence genes, and we can take a look at those, and we're going to take a look at the various types of virulence genes that we have at each of the different stages of pathogenicity. So the first one is adhesion. So, of course, we have portal entry first. It gets into the body. So exposure, and then we have adhesion. So this is where we're going to see those virulence factors come into play. So in the exposure and adhesion steps, we have the adhesins, which we've already mentioned. So these are proteins or glycoproteins that are found on the surface of a pathogen, and that's going to attach to receptors on the host cell. We see these in bacterial pathogens, viral pathogens, fungal, and protozoal. So we can see these in many different types of pathogens. An example that the text gives is this type 1 fimbrial adhesion, and this is actually on the tips of the fimbria, that's why it's fimbrial, of the enterotoxigenic E. coli, the ETEC, and this is what is going to allow those cells to attach to the mannose glycans that are expressed in intestinal epithelial cells. So when we think of E. coli, remember it's it is in the intestines there. And then what they do is they utilize these fimbrial adhesions that are on the tips of their fimbriae to attach to the cells. They can attach to those mannose glycans, and then it allows them to get into the cell. So it's going to allow them to stop, instead of moving down the intestinal tract, it allows them to attach or adhese to the intestinal tract, and then allows them to get in. So that's just another example, since we've already spoken a bit about adhesins. So then what we haven't talked about as much is the invasion step of pathogenicity or pathogenesis, and then some of the different types of virulence factors that are produced for that. We have spoken a little bit. So we have bacterial exoenzymes that we already mentioned and toxins. These are very effective because they pass close to every cell in the body. So once these organisms have attached themselves to the body in some way, then they can utilize these exoenzymes and toxins, which we spoke about the H. pylori and how it can make urease, and that kind of liquefies the mucin gel in the stomach there and allows it to invade further. So they can be very, very effective. And then also, depending on where it is, it's going to be close to every cell in the body. So especially if we're talking about something that produces a toxin in the bloodstream, that can travel through the bloodstream, and that's going to pass by every cell in the body, and that would be very effective then. However, there is a downfall to these types of things. If we're talking about invasion into the bloodstream, is that many immune system cells are in the bloodstream. So if we're talking about invasion specifically in the bloodstream, and we're talking about producing toxins in the bloodstream or attaching to red blood cells, that kind of thing, a downfall there is that that's where we also find many of our white blood cells and other things like antibodies and things. So that's also kind of a downfall there, is that if they're trying to invade via the bloodstream, then they're going to be doing that right by all of the things that are trying to catch them or mark them or do whatever. So that can be a difficult thing. If we have the presence of pathogens in the bloodstream, that's called emia. That was actually on one of the tables that we looked at in a previous section here. So bacteremia is bacteria in the blood, viremia, viruses in the blood, and toxemia is when you have toxins in the blood. And then we also have something called septicemia. People will say that they are sepsis or somebody is sepsis. This is when there is bacteria present and multiplying in the blood. So when there's an infection of the blood, and that's being sepsis or septicemia. is when we have the patient is septic, and this can actually lead to shock. And that's why this is such a big deal, is because it is in the bloodstream. And since it is in the bloodstream and we have multiplication of the bacteria in the bloodstream, then what that means is that, again, this is going by all of the cells of the body, which means that it's a pretty systemic infection, because it's passing throughout the entire body. It's not just in the stomach, for example. And then this can lead to shock. And this shock can be life-threatening, because what happens is, because of this bacteria being all over the place, it's present and it's multiplying throughout the entire body, the body is going to try to do things in response to that. But oftentimes what happens is it goes so overboard on the response, because it's the entire body, that then we have some severe decreases in blood pressure, and so severe that that can lead to shock and then can actually kill a person. It also prevents cells and organs from getting enough oxygen and nutrients. So if the body has this systemic infection, so this infection that, is in the bloodstream, then it is, all of these bacteria are going to be using the nutrients, right? So just like we think about it, using nutrients in our broth, for example, in the laboratory, it's going to be using the oxygen that's present. It's going to be using any of the nutrients, so the ATP, but, anything that comes from the ATP. So it'll be eating, utilizing lactose or glucose or whatever it is that's in the bloodstream and that organism can use. And since it's using all of that and it's multiplying at great speeds, like if we think about E. coli, remember that multiplies, it doubles its population in 20 minutes at the right temperature, which is what we would have in our bodies. So they're utilizing all of these nutrients, utilizing all of this oxygen, and therefore our cells then are not getting the nutrients and are not getting the oxygen. So not only is our body trying to fight off something throughout the entire body, but then also we have the fact that our cells and organs just aren't getting the things that they need to get to function. And so then that's why it can lead to shock and then can actually eventually kill a person. So we did speak a bit about exoenzymes, and we're going to talk a bit more about them now. Remember that they are extracellular enzymes, so outside of the cell, enzymes that enable a pathogen to invade host cells and deeper tissues. So we have a wide variety of targets that are going to facilitate invasion or support its own growth. We did already speak about H. pylori with its exoenzyme of urease, but here's another example here. Hyaluronidase S, and this degrades the glycoside hyaluronic acid, and it acts as an intracellular cement between the adjacent cells in the connective tissue. So if we look at the image down here, we have our hyaluronic acid, which is between the cells, and it, is kind of gluing those cells together, making sure that nothing can get through them or between them to get further into the body. So it's within this connective tissue holding the cells together, and its function is to protect the body. However, because of the way that evolution works, then we have organisms like S.aureus, Pathogenes, and Superantigens that are able to produce hyaluronidase S, which is able to degrade that. So that's one of the ways that they have tried to evade our barriers, our ability to protect ourselves, by being able to produce an enzyme that now degrades that. So then that means that the pathogen is going to pass through the tissue layers at the portal of entry and then disseminate elsewhere in the body. So if, for example, we get any of these bacteria in our mouth, say we had it on food or something, although these aren't typically food ones, say we just breathe in a cough cloud. I don't know if that's better, but we breathe in a cough cloud and that gets into our mouth, say our throat, and then this is going to attach or adhese to the cells in the mouth or in the throat, and then they're able to produce this hyaluronidase S, and then they're going to be able to start to just eat away. You can see the bacteria eat away at this cement in between, and then if this is, say, our throat here, it's going to be able to work its way through and get into the bloodstream pretty quickly. So that's just another example of an exoenzyme. All right, so we are going to now move along and talk about some other bacterial exoenzymes. And the first one is DNase and other nucleases. So these are enzymes that are going to break down nucleic information, or specifically in this case, DNA. So this is going to degrade extracellular DNA, and this is as a means of escape. So an organism that we see that utilizes this is Staph aureus. So at the site of infection, so say you get cut and it had, whatever you were cut with had some Staph aureus on it. At that site, that's where we have infection. So that's where we have the Staph aureus getting into the body and then starting to multiply. We're going to have dead bacteria there, and host cells. And so the bacteria, as they're producing, some of them are going to be burst and they're going to be dying, especially if we have, white blood cells and things that are starting to fight them off and killing them. And we're also going to have host cells that are lysed at the same time, because if there's a cut, then there's, dead host cells as well. And so these dead bacteria and these dead host cells are going to be burst, and all of their contents are going to be falling out all over the place and into the fluid in the area. So their contents are released, and then the pathogen can actually get stuck in the DNA, because remember, DNA is this really big stringy thing, and it is wrapped very tightly around these histones and things to make it more compact. But that is typically when we're utilizing a lot of the histones, and when we have our DNA as being very, very compact, is mostly when it's going through mitosis, so that we can separate the chromosomes correctly for our cells. Now, on kind of a typical everyday basis, our cells aren't that compact, and they aren't that that nice little looking kind of arms and legs thing that we think of a chromosome, the X's. What they are more so is like a big ball of yarn that is not all wound up, but it's kind of all over the place, because at that time, all of our enzymes are going in and they're, making different genes, turning them into mRNA, and then they're going to go out and make proteins. So then when those cells that are just doing their everyday thing get cut and they're lysed open and they burst out, then all of that DNA is like a net. And actually we'll talk about in, I believe, chapter 18, nets or NETs, and these are actually produced by some white blood cells specifically to catch bacteria. However, just on their own, the dead bacteria in the area and the host cells in the area are going to have their DNA kind of in the fluid around, and then those are going to catch other bacteria. Now, S.aureus has developed this ability to have DNase, and it can produce DNase outside of its cell, so extracellular, and then it can utilize this to get itself free. So by degrading the DNA, it's going to release the pathogen so that it can get further into the body, further into the bloodstream, and then continue to spread throughout the tissue and then throughout the body. Another bacterial exoenzyme are the phospholipases. As its name implies, it degrades the phospholipids of a cell, so that's their cell membrane. And it can do this either of the host cell, so once it attaches itself to a host cell, it can then start to degrade that plasma membrane to get inside of the host cell and get to some of those nutrients, or in the phagosome. So remember, the phagosome is when we have a white blood cell. The white blood cell grabs onto the bacteria and brings it into itself through phagocytosis. And inside of that white blood cell, when it's come in through phagocytosis, it's called a phagosome, which is just pieces of plasma membrane, a plasma membrane bubble. So if an organism is able to produce a phospholipase, then what it can do is it can degrade that phagosome, and then it can be free from that white blood cell so that it can then, destroy that white blood cell, use its nutrients, and then get free and get to the rest of the body. So this is when it degrades the host cell membrane to access the interior of the cell, or it can do that in the phagosome in order to get free. An example is phospholipase C. So Bacillus anthracis is able to produce phospholipase C. And then, so when Bacillus anthracis is digested by a phagocytic cell, it's brought into via a phagosome, then this enzyme is able to degrade that phagosome. It's released into that white blood cell, and then it's able to immediately start to multiply because it's utilizing the nutrients of the cell. So phospholipase C is an example of that that I've already described. Another example of a bacterial exoenzyme are different proteases. So proteases are enzymes that digest proteins. There are several different types of proteases, and typically they're classified according to their target, so their substrate target, or if they contain metals in their active site. So this is why we need vitamins and minerals, for example. We need those minerals in very, very small amounts and trace amounts. But we need them for proteases within our body, but then also different pathogens need minerals so that they can utilize them in their proteases as well. So for example, serine protease, it's going to degrade proteins that have the amino acid serine. So it is not able to degrade other proteins or break down other proteins, it has to break it down at the serine. So if that amino acid serine is present, then it can utilize that protease. Another one, zinc metalloproteases. As their name implies, they contain zinc, which is necessary for its enzymatic activity. It's necessary for it to go in and break down these proteins. Another example is collagenase. Collagenase is going to destroy or digest collagen. Remember, collagen is our connective tissue. We have a lot of collagen between our cells. We have a lot of collagen that are connecting different types of tissues together. So this is a protein that's within these connective tissues, and then it's if an organism is able to make collagenase, then it can make its way through those tissue layers. So in the image here, we have Clostridium perfringens here, and it's allowing, we're in the bloodstream here, the lumen of the blood vessel, and then it is able to produce enzymes to break its way through here, and then it's going to be able to get into other tissues that way. So we have our endothelial cells, and then we have our connective tissue, and within our connective tissue we have collagen, and so it's able to degrade that collagen and then get further into the body. So then we can talk about our bacterial toxins, and we haven't really spoken much about our endotoxins. So toxins, again, are biological poisons. They're going to assist in their ability to invade, the ability for the pathogen to invade, and then it causes damage to tissues. So we have a term here, toxigenicity. This is the ability of a pathogen to produce toxins, just like pathogenicity. It is the ability to do that. And then there are different levels of that, so different severities. So we have our endotoxin. When we're talking about our endotoxin, what we're talking about is lipopolysaccharide A, or lipid A. So our lipopolysaccharide is in our gram-negative bacteria. If you remember back to the structure of our cells for our gram-positive and our gram-negative bacteria, recall that our gram-negative bacteria had that additional layer on the outside of its plasma membrane. And remember that that additional layer was our lipid A layer. Now we're going to look at it in more detail because this now is an endotoxin. It was before, but now we're going to look at it in terms of an endotoxin. So in our gram-negative bacteria, we have our lipopolysaccharide that's within that plasma membrane. And we can actually see here that these are our kind of phospholipids here, which contain lipid A. And then this has a core to it, and then there's this O antigen. Now, this is on the surface of our gram-negative bacteria, but when our body goes in with its white blood cells and it starts to destroy gram-negative bacteria, or when we target it with antibodies and things like that, then it releases this lipopolysaccharide when it is lysed by our white blood cells, for example, or destroyed, or when it's dividing. So if it's dividing, it is kind of making new cell wall and new plasma membrane. And so then we have this production of this lipopolysaccharide. So as it's dividing, it's going to be releasing this endotoxin, which is why it's a toxin, is because if that bacteria is in our body and it's multiplying, then it's multiplying and releasing this toxin. But even if our body goes in and it's able to identify the infection and the bacteria and get it and start to break it down, then it can also release it because it is lysed. What this does then is it triggers the immune system's inflammatory response. So this is going to cause the whole body to become inflamed, or the area, I guess, the site of infection, if we're not talking about septicemia. But the immune system is going to be triggered in this case. So this is going to trigger the immune system. At low levels, it may help the host, because if we have very low amounts, likely what's happened then is we're just starting to have an infection. And so the body, once that happens and it triggers the inflammatory response, that inflammatory response is actually helpful for our body. We want the inflammatory response. That means that our body's working, and it's working to fight off the infection. However, at higher levels, then it can cause things like organ failure and shock and things like that. So this is if we have a septic situation where we have the bacteria in the bloodstream and it's multiplying rapidly. Every time it's multiplying, it's releasing this lipopolysaccharide, which is an endotoxin, and it's very toxic to the body, and it's going to cause the body to respond, and when it does, it responds with the inflammatory response, and the entire body then is going to have this huge response of inflammation, which is an attempt for our body to get rid of the bacteria, and we'll talk more about that in chapter 18. It's a response for our body to get rid of the bacteria, but it can also start at high levels to destroy our own cells, so it can be very damaging. So if we take a closer look at this lipid A, the lipid A piece here remains stable at high temperatures. So at 121 degrees C is where it has to be in order for it to be destroyed or to be inactivated. So if we think about 121 degrees C, remember that that is where we have our autoclave set. So when we're trying to kill bacteria, we can kill our gram-negative bacteria at that 121 degrees C. And that's how we can inactivate that endotoxin. But it remains stable at high temperatures, which means that even inside of our bodies, even if we have a fever, then we're not going to be getting rid of the gram-negative bacteria. We're not able to inactivate. We might be getting rid of the gram-negative bacteria. Let me take that back. But we're not going to be able to inactivate this lipid A. We have different ways that we can test how much of this we have in the body, or in a sample rather. And we do this by utilizing the LAL test, which is the Limulus amoebocyte lysate test. This is going to tell us in a sample how much of this lipid A that we have. So in this test, what they do is they take blood cells of the horseshoe crab, which are blue, which is very interesting, the blood cells of the horseshoe crab, and then they mix that with the patient's serum. So they do a blood draw, they get the serum from there, they mix that serum with the blood of the horseshoe crab, and then it causes a color change. So it changes from that bright blue, and then it also is going to coagulate. So it's going to become gel-like, and it'll agglutinate, which is attached to each other and kind of stick together. And so then that's how we can tell with the amount of color change and with the amount of coagulation, one, if it's present, and two, somewhat to a degree, what kind of degree we have. It's not accurate, of course, or it's not very specific, of course, but it gives us an idea of how much endotoxin is inside of a person's body. We also have something called an ELISA, which is an enzyme-linked immunosorbent assay, ELISA, and this is going to use antibodies to detect the endotoxin, and we're going to talk about how we can use antibodies to detect certain things, including to detect this endotoxin. We can identify the lipid A, and this is just another way for us to determine if a person has an infection by a gram-negative bacteria, which would be important because if we can distinguish between gram-positive and gram-negative, then we have a chance at using a correct antibiotic if they choose to go that route. So using an ELISA is one way, using the LAL test is another way to determine if there is this endotoxin within the body. So that was the last endotoxin. Now we're going to be talking about exotoxins. So endotoxins are the ones that are produced within the cell. And then we have our exotoxins. So we produce exotoxins, or bacteria produce exotoxins, in typically all types of bacteria, but mostly gram-positive. So when you think about endotoxin and exotoxins, a lot of times when you think about endotoxin, what you're really talking about is the lipopolysaccharide. And that's that lipid A that we find in gram-negative bacteria. So the endotoxin is in gram-negative bacteria. Exotoxins are ones that we typically see in gram-positive bacteria. So these toxins are more specific. The lipopolysaccharide, or the lipid A, is in all gram-negative bacteria. That's part of their cell wall, part of their plasma membrane. And the exotoxins are going to be more specific, and so different types of gram-positive bacteria are going to have different types of exotoxins. So they're more specific in their action and in the cells that they interact with. So they are going to be interacting with those receptors. They are heat labile, which means that they're denatured at temperatures above 41 degrees Celsius. So remember that our body is typically around a 37 or so degrees Celsius. So if we increase our body temperature a bit, then we can actually start to denature some of these exotoxins, which is different than the endotoxin, which we're not able to really do with a fever. However, very small concentrations of exotoxins can be lethal. So endotoxins, while the lipopolysaccharide can cause, organ failure and shock, it has to be at extremely high levels, which does happen, but it has to be at extremely high levels. Low levels can actually help our immune system to fight something off. However, with exotoxins, the very small concentrations can be lethal. So, for example, botulinum toxin has an LD, remember, a lethal dose, median lethal dose, of 0.000001 milligram per kilogram, versus the endotoxin has an LD50, a lethal dose 50, or a median lethal dose, of 0.24 milligrams per kilogram. So, our exotoxin is a whole lot more lethal than our endotoxin. Down here at the bottom, we have a comparison between endotoxins and exotoxins that are produced by bacteria. So again, our endotoxin is from our gram-negative bacteria, whereas our exotoxins are mainly gram-positive and some gram-negative bacteria. So we can say for sure that the gram-negative is an endotoxin, but for the other one, some gram-negative are also able to produce exotoxins. The composition, a lipid A component of the lipopolysaccharide that we find in a gram-negative bacteria. Over here, though, our exotoxins are typically proteins. The effect on the host, so we have our general systemic symptoms, so we have inflammation and fever, kind of our general overall response to a toxin. In our exotoxin, we have specific damage to cells. So it depends on this receptor-mediated. That's why we said that these are more specific in their action. So receptor-mediated targeting of cells, and then they have specific mechanisms of action. It's not just initiating this inflammatory response. Heat stability, so our endotoxin, again, is very heat stable. Over here, our exotoxin is not very heat stable, but some are. The LD50, remember the LD50 for an endotoxin is high. The LD50 for an exotoxin is low. So some exotoxins. The first one is called an AB exotoxin. So it's an intracellular targeting toxin. So it's called an AB exotoxin because it has two subunits, A and B. So the B subunit is responsible for cellular specificity of the toxin, and it initiates binding to a specific cell surface receptor. So if we look at the image down here, we can see the different subunits. It's an AB exotoxin, so we have our A subunit and our B subunit, and you can see that the B subunit is responsible for interacting with the cell membrane on the cell that it's trying to infect. So we have our cellular receptor that's actually on the cell membrane of the cell here, and that's going to interact with our B subunit. You can see it's kind of a lock and key mechanism there. So then what that does is it attaches to the cell membrane, and then that's going to tell the cell to bring this in via endocytosis into a vacuole. So it's going to initiate endocytosis. So you can see our AB is now coming in via endocytosis. This is going to merge here, and then we are going to be inside of the cell in a vacuole. Once it is in the vacuole, the A and B subunits are going to separate as that vacuole acidifies. So that's something that happens. This acidification of the vacuole is something it turns it more acid so that it can start to break down the pathogen or the toxin in this case. So as it's acidifying, though, that is going to break this bond between the A and the B subunits. So then the A subunit is going to be able to get out of that vacuole and enter into the cell's cytoplasm out here. And then what it's going to do is it's going to interfere with its internal function. And so that's how it works as a toxin, is the A piece, the A subunit, is what's actually going to start to interfere with specific internal functions of the cell. Whereas the B piece just stays in here in the vacuole, and it just stays attached and it gets degraded and broken down. But that's how the AB toxin was able to get into the cell to begin with, is because it had the B subunit, and then they're able to separate, and then the A is able to go around and destroy things and cause problems, while the B subunit then just already did its job and is going to stay there. So we're going to look at an example of an AB exotoxin here with the one that Corynebacterium diphtheria is able to make. In this case, you can see we have our B subunit has already attached to the receptor and has been brought into the cell, and we have our A subunit here is already detached. So it's detached, and then what it's going to do is it's going to facilitate the transfer of this ADP-ribose onto the elongation factor protein on the EF2. So if you look here, it utilizes this NAD, and it's going to take this ADP-ribose, which is this kind of green pill-looking thing, and it's going to transfer this ADP-ribose onto the elongation factor protein. So it's going to take this and it's going to stick it into this protein. What this does is it's going to inhibit protein synthesis in the host cell. So instead of an amino acid coming along, so here's our growing protein, instead of an amino acid or a tRNA coming along here, remember we have our tRNA coming along, and then what's attached to it is it's an amino acid. Instead of that, which then can form a peptide bond to the protein, instead we have our ADP-ribose gets stuck in here, and then it stops transcription. And so then the whole thing is going to fall apart, and we no longer have protein synthesis, and that's going to stay stuck there. And so then as this toxin keeps doing this, taking these ADP-riboses and putting them into this process, then it's going to be stopping all of these ribosomes from doing what they're supposed to be doing and making proteins. And then as they're no longer making proteins, then that cell is going to die because it can't make its enzymes. If it can't make its enzymes, it can't function. So the next example we're going to look at is botulinum toxin. So we're going to look at a couple of different neurotoxins. This one comes from C. botulinum. This is also what is utilized for Botox. So if you're familiar with or have heard about Botox and its various uses, what we're talking about doing here is utilizing the botulinum toxin and using that as an injection. So the way that this works is that we have our B subunit. Our B subunit is going to bind to the neuron, and then it's going to allow entry into the neuron at the neuromuscular junction. So if we look at our organism down here, then what we're doing is here's the muscle cell. This is the cytoplasm of the muscle. And then this is our neuron here because we're talking about the neuromuscular junction, so the junction between the two. And then our B subunit is going to attach to this neuron at the neuromuscular junction. Then what happens is that the A subunit is going to act as a protease. If the A subunit is acting as a protease, remember that's something that breaks down proteins, so it's going to cleave the proteins that are related to acetylcholine release. So in here we have acetylcholine in these vesicles. Now if our A subunit is going to go in and it's going to cleave these proteins, what it's doing is it's going to say, you know, these little vacuoles of acetylcholine, typically what they do is they go to the edge of the cell, they release their contents into that neuromuscular junction here, then they're able to attach to the receptor and it tells the muscle cell to move. Now, over here with the botulinum toxin, it's saying it's preventing, utilizing this protease, preventing the release of acetylcholine. So if there's no acetylcholine release, then that means that there's no acetylcholine in that neuromuscular junction that's attaching to the receptor. And if that's the case, then there's no muscle contraction. Because over here, remember what it's doing is it's telling the cell to contract. So in this case, what it's doing is it's staying relaxed. So if there's no communication from the nervous system to tell the muscle to contract because no acetylcholine is released, then that muscle doesn't contract. That can be a huge problem because we have some muscles we really want to be contracting, like our lungs are surrounded by muscles, meaning our intercostal muscles, which help us to breathe, and our diaphragm, which help us to breathe. So our lungs don't have muscle, but the muscles surrounding them are all required for breathing. And so our diaphragm muscle, then if we get the botulinum toxin near those neurons that are connected to the diaphragm or closely connected, then our diaphragm muscle will stop working. If it stops working and it's just relaxed all the time, then that stops us from breathing. Also our heart muscle, right? So if we have our neuromuscular junction there, we want our heart muscle to keep pumping our heart. But if we have botulinum toxin that's preventing that release of acetylcholine, then our heart muscle can stop as well. So both of those, of course, can then cause death because we just have a constantly relaxed muscle, including heart muscles and the diaphragm and intercostal muscles.
So the next exotoxin that we are going to talk about is also a neurotoxin, and that is the tetanus toxin. So the tetanus toxin works in similar ways that we just saw to the botulinum toxin, but a little differently. So in the case of the tetanus toxin, this is going to bind to inhibitory interneurons. So if we take a look at the image down here, we have our normal mechanism here. We have the cytoplasm of the muscle and the muscle cell. We have our receptors. This is our neuromuscular junction. So we have our axon terminal here. This is the axon of the neuron that's going to stimulate the contraction of the muscle cell or initiate the contraction of the muscle cell. Remember, that's done by releasing acetylcholine via these vesicles. Now over here, we have our inhibitory interneuron. Our inhibitory interneuron is what is going to communicate to the neuron. You can see here via the release of glycine and GABA. It's going to release the glycine and GABA, and that's going to tell this neuron here to not initiate the release of the acetylcholine via the vesicles into that neuromuscular junction. So in the case of our tetanus toxin, this is when we have our abnormal mechanism. So the tetanus toxin is going to prevent the release of the glycine and GABA over here, and it's no longer going to go into the junction here, initiating or attaching to the receptors here to tell this neuron, the original neuron, the excitatory neuron, to release this acetylcholine into the neuromuscular junction. So then what happens is that this neuron, this original neuron, this excitable neuron, is never going to be shut off. So it's going to be constantly releasing acetylcholine into that neuromuscular junction. When it's doing that, then that means that we always have the acetylcholine attaching to these receptors. If we always have the acetylcholine attaching to the receptors, that means that this muscle is constantly contracting. So up here it inhibits the release of the interneurons, and we have permanent muscle contraction. So the first symptom is stiffness of the jaw, or what's called lockjaw, and this is the first time that we kind of it can be an indicator that there is tetanus toxin in one's body, is if we have this stiffness of the jaw or lockjaw. And this is because of that constant release of acetylcholine is going to constantly keep those muscles contracting. We also have violent spasms because, of course, we're constantly releasing acetylcholine, constantly telling our muscles to contract, so we have spasms. This can lead to respiratory failure because, of course, again, if we have our diaphragm and our intercostal muscles and we're constantly telling those muscles to contract, then they're not going to be relaxing at any point and allowing for the lungs to expand and then to contract and then expand and then contract. So we'll have respiratory failure, so an inability to breathe, and then eventually death because, of course, if we are constantly telling our muscles to be contracting and never relaxing, that also includes our heart muscle, as well as, we just mentioned, the muscles in the thoracic cavity, the diaphragm, and the intercostals, eventually leading to the inability to breathe and the inability for our heart to pump as it needs to. So then the next exotoxin we're going to talk about is kind of in general what are called superantigens. So superantigens are exotoxins that trigger an excessive response. So an excessive, non-specific stimulation of immune system cells. Now, what this does is it causes them to secrete lots and lots of cytokines. And cytokines are these little chemical messengers. When we have a production of cytokines, what it does is it produces our immune system response. So we have our stimulation of the immune system cells. They secrete cytokines, which is going to increase the immune system response, but it's also going to cause an inflammatory response. So these cytokines, when they're released, is actually something that our body does that's helpful, and we'll talk about these things in Chapter 18. When we release the cytokines, it's telling the rest of the immune system to come help, It's also causing inflammatory response, which is meant to help the body to fight off any pathogens. Now, when we have a superantigen, this causes, again, an excessive, non-specific release of these cytokines. And since it's excessive, then this causes an excessive immune response, an excessive inflammatory response. Oftentimes, this leads to life-threatening high fevers. Part of the inflammatory response causes fever, and if we have an excessive inflammatory response, that means we have an excessive fever. So that can be life-threatening high fevers. It also dips the blood pressure because of the response, because of this inflammatory response. We actually have a dilating of the blood vessels, which causes a drop in the blood pressure, and that can be so low that it can also cause problems as well, life-threatening. Also, we have multi-organ failure. Again, because of this inflammatory response, our body is going to start to shut down because it's not getting the blood that it needs, it's not getting the nutrients it needs, it's not getting the oxygen that it needs, and we start to have organ failure. This can lead to shock because when our organs are failing, our body goes into shock, and then eventually death. So superantigens are incredibly, incredibly dangerous. The most well-known, I believe, superantigen is toxic shock syndrome toxin. This is produced by S.aureus, and then mostly this happens in vaginal toxic shock syndrome. So if anybody's ever heard on commercials or anything for feminine products or seen the little pamphlet that comes in the feminine product boxes, they talk about toxic shock syndrome. And this is when that product is used for too long or kept in for too long, and then that can cause toxic shock syndrome. And that's because of a buildup of the Staph aureus. Once they get to a certain level, via quorum sensing, they're going to start to produce this toxin. When they produce this toxin, it is a superantigen. So a person can go through and be just fine and just fine and just fine until all of this Staph aureus starts producing the superantigen. Then we have this excessive immune response, inflammatory response, high fevers, the low blood pressure. It can have organ failure, shock, and death. And this can all happen very, very quickly. So within hours, within a couple of days, this can happen to where a person can die, even though they seem to be otherwise healthy.
All right, so next we're going to take a look at some virulence factors for survival in the host. So the first one that we're going to take a look at are capsules. And we've already taken a look at these, specifically when we've taken a look at the structure of bacteria. But recall that the bacteria uses capsules to be more resilient in order to evade the immune system and also to allow it to stick to certain things. So it allows it to stick to things because it's sticky, made up of those glycoproteins. Remember, glyco is carbohydrate. And if you think of sugar, you can remember that that's very sticky. So the capsule is very sticky, allows it to stick to things inside the body. But then also, as you can see in the picture here, you can see this big, thick capsule. So that would then make it more difficult for our body to get rid of or kill the bacteria because it has to work its way through the capsule. So its composition actually prevents our immune cells from grabbing onto it. So you can see here in this middle image here, we have our phagocytic cell, and then we have the antibody. That's typically what is going to interact with the antigen, which is the part that tells us when it's a cell that we don't want in the body. It's non-self. And so our phagocytic cell isn't able to attach its antibody to the antigen to then initiate the phagocytosis. And so since it's not able to initiate the phagocytosis, then it kind of goes by it. So the capsule then protected that bacterial cell. So it also makes it larger. So even if the phagocytic cell is able to understand or recognizes the fact that it's not a self cell and it shouldn't be in the body, it also is very difficult for it to grab onto it and bring it into itself, into that phagosome. So it prevents its adherence for phagocytosis, and then also its larger size makes it really hard for it to do that. So for example, S. pneumonia is more virulent when it's encapsulated. So we can have things like meningitis or septicemia and some other more severe things when it's encapsulated, but it's not always encapsulated. And so when we have non-encapsulated bacteria, even S. pneumonia, then we don't have as much of an issue with that. We don't have things that end up being life-threatening oftentimes. And you can see the difference really between this non-encapsulated bacteria here and then this really thick capsule in the image here it clearly protects that bacteria. The second one is proteases. So proteases are going to attack and digest the antibody molecules. So we already mentioned here that our phagocytic cell utilizes an antibody to attach to the antigen or the non-self cell. Now, when an organism, when a bacteria, for example, is able to make proteases, you can see in this picture over here on the right, or this illustration, if it's making proteases, it can actually break down these antibodies. Because, again, proteases go in and they cleave those bonds between those amino acids, and if it does that, then it's breaking that apart. So it can attack and digest the antibody molecules that have attached to the pathogen. So, and we're going to talk about this more in future chapters, but there are two ways that our body is going to utilize antigens that the body is going to utilize antibodies. One, as we just noted here, is with the phagocytic cell. It's going to recognize it and then engulf it. So even if this bacteria here is making proteases, it can break down this antibody and then it can prevent it from going around and attacking other bacteria. Over here, this is an individual antibody, so one that's not attached to a phagocytic cell, which also happens, and we'll talk about why. But then if this actually does attach here, then the proteases that are made can break it down, and then it will no longer be useful as an antibody inside of our body, and along with the host immune system. So our M protein is another one. So M protein is in the fimbriae of some Streptococcus species. What this does is it alters the surface of it. And again, if they're altering the surface of it, then we have issues with phagocytosis. So our phagocytic cells aren't able to attach onto it, aren't able to grab it and bring it into the cell to be able to digest it or lyse it. M. tuberculosis, Mycobacterium tuberculosis, has a waxy mycolic acid layer that resists some killing mechanisms. So another one is that mycolic acid. Remember at the very beginning of the semester when we talked about gram-positive and gram-negative, and then we also had those that had that mycolic acid layer on the outside, so those mycobacterium. So that waxy layer is just one of those ways that a bacterium can prevent itself from being attacked or being broken down more easily. So we have the M protein, we have the mycolic acid layer, then we have coagulase. Coagulase coagulates things, meaning that it causes clotting. So what this does is it creates a clot around the bacteria. Because when the bacteria, so it causes clots in blood, when the bacteria gets into the bloodstream, what it can do is it can actually utilize the host's the host's blood in blood cells inside of the bloodstream there, inside of the vessels, and it can create a clot, and then that bacteria is just going to hang out inside of that clot. When it does this, however, then our phagocytic cells, again, are going around trying to find these cells that are not self, but this cell has surrounded itself in self cells. So it's not going to be able to be recognized by our phagocytic cells because it doesn't see that there's a bacteria in there. When it's bumping into this thing, all it touches are these red blood cells that have been coagulated and created this clot around the bacteria. And we see this happening with Staph aureus, that it is going to create this. It produces coagulase that coagulates the blood around it and creates this kind of capsule, as it were, of blood cells. Then we also have kinases. So we have specific types of kinases, or general ones we can mention, which are staphylokinase and streptokinase. As you can imagine, they're in Staphylococcus species and Streptococcus species. What these do is they trigger the digestion of blood clots. So not only can a bacteria produce coagulase to cause clotting around itself, but then it can produce kinases, staphylokinase or streptokinase, in order to break blood clots down. So perhaps once this cell, let's pretend a cell gets into the bloodstream, it produces coagulase to create kind of this capsule around itself of blood cells so that it can evade the immune system. Once it gets to where it wants to be, then it can utilize a kinase and then break down those blood vessels, or blood cells rather, and then get into wherever it's trying to get into, so whatever cell it's working its way through. Also, this is useful even separate from coagulase. So even if a cell doesn't actually produce coagulase, it can still produce some kinases, and this is useful for those bacteria if they do get trapped in a clot. So for example, if we get a cut in our body, and then that's how the bacteria gets into our body, as our body is going through the process of clotting to protect that area, prevent blood loss and all of those things, a bacteria might get stuck in that clot. But if it can produce one of these kinases, then it can eat away that blood clot and escape and then get back into the body, or get into the body to start with. So, for example, again, Staph aureus produces staphylokinase. There's a Y in here, staphylokinase. So our Staph aureus can produce the coagulase. It can also produce the staphylokinase, but it doesn't have to have both. So another thing that we see as far as virulence factors is something called antigenic variation. In antigenic variation, what we have is this change in the surface proteins, or an alteration of the surface proteins, so that this pathogen isn't recognized by the host's immune system anymore. So we'll talk more about the immune system in further chapters, but we have our first exposure to a pathogen, and in our first exposure to a pathogen, our body learns that pathogen. So what it does is it builds up an immune system response specifically to that pathogen, so that when we are exposed to that pathogen a second time, when we have a secondary exposure, then our body can go through the process of attacking that and overcoming that particular pathogen much faster, because we have built up an army of cells that can then respond quicker to a secondary or tertiary, or etc. Exposure to that pathogen. Now, when a pathogen is able to have antigenic variation, however, it can get into the body in one form, and then the body will do what it does. It builds up this immune system response so that if it is exposed to that same pathogen again, that it can host a quicker, stronger response. However, particular bacteria, so for example, Borrelia burgdorferi, it can go through the process of antigenic variation where it can alter the surface proteins. And if it alters the surface proteins, our immune system no longer recognizes it as that particular pathogen. It thinks it's a brand new pathogen. And so our body has to respond as it is a new pathogen, because the outside area has changed, so it looks different. And then that means that we don't have as quick or as strong of an immune system response. So we'll get sick all over again. So this happens, for example, in Lyme disease, which is why Lyme disease can be kind of a lifelong condition, or very difficult to get over. What happens is Borrelia burgdorferi can go through genetic recombination when it goes through DNA replication. So this is even within the person. So it's not like you get sick with something, you get rid of it, and then the next time you are exposed to it via another person, that it has changed. This is inside of the body as well. That can happen, but it also can happen just within the original host, or one person. And this is because as it goes through DNA replication, just to go through its simple binary fission to make copies of itself and to grow, we have this genetic recombination. What this does is it causes changes in these genes that are going to code for these surface proteins. So as it's going through DNA replication, it actually changes its genes, and that changes the surface proteins. And so then the body, even though it's still in the body, and maybe the body has responded to the first version of Borrelia burgdorferi, for example, and it's fighting that, and it's doing a really good job because we've built up this immune system to do that. Now, even inside the same human, as it's going through the process of dividing, it actually makes different versions of itself. And this is why we have chronic Lyme disease, and why it's so difficult to get rid of Lyme disease is because every time it goes through the process of dividing, which we know is very often, it can change the way that it looks, which means that the body is constantly trying to fight off what it sees as a new pathogen. This is another example here. Neisseria gonorrhea has antigenic variation as well, and it's type 4 pili. So that's also why we have some illnesses like gonorrhea that you have forever, or seemingly have forever, because of this changing that's constantly happening. So even if the body is healthy, even if you keep the body healthy, it tries to fight it off, but it changes the way that it looks every time it divides, or perhaps every time that it divides, and then our body is constantly trying to fight what it sees as a new pathogen, and we just never get rid of the disease. All right, so some virulence factors that are not related specifically to bacteria, but are related to viruses. are viral adhesins. And we already spoke about adhesins, so it's very similar. So these are surface proteins within the capsid or the membrane envelope. So remember, sometimes we have enveloped viruses and sometimes we have naked viruses or unenveloped viruses. So we can see that in the membrane envelope if it's enveloped, or within the capsid if it's not enveloped. And then these surface proteins are going to allow them to bind to specific host cells. So just as we already spoke about adhesins, working the same way but just with a virus. So some of these are called spike proteins. So for example, spike protein hemagglutinin on the influenza virus. So the flu virus allows for attaching to the respiratory cells and intestinal cells. So they're specific. Right, so we don't have the influenza virus trying to attach itself to the liver, for example, or trying to attach itself to the kidney or something. It doesn't cause kidney infection per se. It can cause some issues with the kidney if we have other issues going on. But what it's going to do is it's going to attach to specific receptors in the respiratory and the digestive system. Another example is with HIV. Remember, we have the glycoprotein, which is GP120, is going to attach to that CD4 receptor on our immune system cells, on our CD4 immune system cells. So that protein spike is going to attach, the protein spike is called the glycoprotein, GP120, is the one that's going to attach to that CD4 receptor. And then there's that secondary interaction that causes a shift, and that's between the CCR5 or the CXCR4. And then once those two things are attached, then we have entry into the cell. It's able to get itself into the cell that way. So those are examples of different viral adhesins, so allowing them to stick to the cell, and then they're able to invade after that. And then some other things that we see with viruses are antigenic drift and antigenic shift. With antigenic drift, what we have, the reason it's called drift is because it's just drifting a little bit. It's a slight change. And what this means is that we end up having point mutations in the viral DNA or RNA. And what this does is it causes slight changes in the spike proteins, the hemagglutinin and neuraminidase. So these spike proteins, again, are the ones that are going to be on the outside of the virus. And so if we have point mutations, remember in a point mutation we can have just very small changes. So it might change one amino acid, and so then it folds slightly differently. And if that's the case, inside of the virus itself, it just continuously does these little tiny antigenic drifts, these little tiny point mutations. And this is the first image here. Antigenic drift is where we have virus A here, and then we have our two different spike proteins here with the yellow and the green, and you can see their different shapes. And then when we have this point mutation, which they're indicating with this red change in the DNA, then you can see that there's a shift in the way this hemagglutinin spike protein looks. So now it's mutated, it looks slightly different, and again, then that means our immune system thinks it's an entirely different pathogen and has to go through the process of making an entire army to get rid of that particular pathogen. Secondly, here we have our antigenic shift. So in this case, this is when we have a major change in the spike proteins. So that's why it's a total shift rather than just a slight drift. So a major change in the spike proteins, and this is due to gene reassortment. So over here we have an image that's showing antigenic shift. And so we have virus A and virus B, and here I have mentioning that it's typically when we have two different influenza viruses. This is when we often see this change, which is why every single year we have different types of flu going around. So if you get a vaccine for flu, it's just good for hopefully that 1 year, and then it doesn't make any difference whether you had that the previous year or not because it's changing. And this is one of the reasons that that happens. So, or one of the ways that that happens. So if we have virus A, we can pretend that's flu virus A, and then we have virus B, which could be another flu virus B. And then inside of the host cell, remember these are obligate intracellular organisms, so they can infect the same cell. And if they're infecting the same cell, then what we can actually end up seeing is that they're going to start to exchange genetic information inside of that host cell. And so when they're going through the process where they break their capsid open and then they start to take hold of the ribosomes and other machinery, both of these viruses will be doing that. And since they're both doing that, sometimes they can mix up their DNA as they're repackaging their capsids and bursting out of the cell. And so we have both of those going in. You can see that we have different spike proteins here. So you can see the different colors, different shapes. And then you can see that we now have virus C. First, we had virus A and virus B, and then now we have a totally different one, which is virus C. And that's because we have a mix. Now we have the hemagglutinin from virus B, and we have the neuraminidase from virus A. And so now that's a new organism. So when our immune system tries to recognize it, it might host, or it might be able to launch a slightly larger attack on that one because it perhaps has recognized those specific spike proteins from the other viruses if it was already fighting those off. But in this case, say virus C now goes into a whole new person that didn't have virus A or B, and now it's a completely new virus. Our rate of antigenic variation in influenza viruses is high, and that's why these vaccines are given every year, is because this does happen, and it happens very often within each individual person. This can happen, and then we end up creating new viruses, or they end up creating new viruses as we transmit the virus along to new people each time.